My name is Anthony Mustoe, and I'm a 2015 Arnold O. Beckman Postdoctoral Fellow. It's been an outstanding honor to be a member of this great class of scientists, and I'd like to take this opportunity to tell you about my postdoctoral research. My research focuses on understanding how RNA structure influences gene expression, specifically of mRNAs or messenger RNAs. Messenger RNAs typically serve as the message that conveys information from DNA into protein. And so while we typically think of these as simple messages, I think that this view glosses over a lot of the very fascinating and important regulation that occurs at the level of the messenger RNA itself. I like to use the analogy that mRNAs are a lot like emails. So emails have two essential components of information. The first is the header, and the second is the message. And when we write an email, we typically primarily think about the message, which contains the primary information of the email. But if you think about it, the header also contains really essential instructions. So who should this email go to? For example, what the subject is has an important role that plays in whether we read an email or not. And so the same thing happens with mRNAs. The core message itself is what's made into proteins, but the header contains information about how much protein to make. Should we make 1 copy, 10 copies, or 100 copies? And where and when should we make this protein? We are just beginning to understand these questions. My research focuses specifically on how the structure of mRNA molecules contribute to their regulation. All RNAs, including mRNAs, fold back on themselves into complex structures. These structures in turn play diverse regulatory roles, such as serving as protein binding sites or sequestering or exposing specific regulatory sequences. My postdoctoral advisor, Kevin Weeks, pioneered several technologies that have just recently made it possible for us to study mRNA structure. These so-called chemical probing technologies use a chemical probe that will react with flexible RNA nucleotides. We can then use next generation sequencing technology to read out those sites of chemical modification, and those reactive nucleotides indicating flexible nucleotides, whereas unreactive nucleotides are base paired. A core project of my postdoc was using these chemical probing technologies to determine the structure of mRNAs in the E. coli bacterium. This research led to 3 pretty striking findings. The first of these is that when we looked at mRNA structure across different genes, it varied dramatically, suggesting to us that structure can be playing really diverse roles in regulating gene expression. The second core finding was that mRNA structure seems to be playing a very central role in controlling how much protein is made from each gene. So this finding has really important implications for biotechnology applications, where we're, for example, trying to design new genes that make a very specific amount of protein. The third finding that was really exciting to us was that we found numerous highly conserved structures that are present in other bacteria. And so we think that these structures are also likely playing important roles in controlling the bacterial gene expression. We are very interested in looking at what the actual functions of these are doing in the future. This is just an illustration of the exciting new opportunities and avenues of research that our work has opened up. The second core focus of my postdoctoral research has been on developing new technologies to further improve our ability to determine RNA structure, which I think nicely aligns with Dr. Beckman's legacy of enabling scientific discovery through technological innovation. One of the core challenges of a chemical probing experiment is that we aren't directly measuring the RNA structure, but rather we're inferring it based on the chemical reactivity of the RNA molecule. And so this inference problem can be very challenging for some RNAs. The approach that I've developed detects correlated signals in the reactivity in an RNA molecule. And from these correlated signals now we can directly tie that to RNA interactions. And so this information substantially improves the sensitivity and accuracy of the RNA structure determination problem, and I think is going to play a very important role further improving our ability to determine RNA structures in the future. One of the cool aspects of this experiment is that in order to enable this correlated chemical probing, we had to change the pH of the reaction, which is directly linked now to Dr. Beckman's legacy as the inventor of the pH meter. Dr. Beckman left an incredible legacy. From the invention of the pH meter to the spectrophotometer, his inventions continue to shape how science is done today. And by establishing his foundation, he's provided an enduring gift to society and the scientific community at large. The Arnold and Mabel Beckman Foundation, by funding scientists like me, has truly played an incredibly important role in funding the next generation of scientific discovery. For me, this fellowship has been more than just about research, but an opportunity to interact with like-minded scientists at the annual Beckman Symposium and to plan out the next stages of my career. Dr. Beckman's lessons for success, including a willingness to take scientific risks and to not be afraid of failure, are lessons that I've really taken to heart, and I'm going to try to keep at the forefront of my mind as I move on to the next stage of my career. So I would like to take one more chance to thank the foundation, the Arnold and Mabel Beckman Foundation, and those who selected me for this incredible opportunity. And I will forever be grateful for the opportunity to continue Dr. Beckman's legacy.